You know, so that itself, I think, is a good example of oligotroph.
So I welcome everyone. So, dear participants, today we are fortunate that Dr. Prakash Salami sir, Dr. Prasun sir, expert present with us. So, I welcome everyone to this roundtable discussion on the topic oligotrophs. So, we all know that this one is the unique group of the microorganisms that survive in an environment where the nutrients are extremely scarce and, for example, in the deep sea ocean water, in the arid deserts and the high altitude regions and the nutrient poor soil also. So unlike the copiotrophs that flourish in the nutrient rich condition, these oligotrophs are specialists, we can say, that survive in the very low nutrient conditions. Means they can survive under the limiting conditions. So these adaptations include the means how they can survive in the low means nutrient conditions. So they have adapted for the slow metabolic rate, high affinity nutrient transport systems that capable of restoring the minute concentration of the organic and inorganic compounds and specialized enzymes that enable them to break down complex molecules into the usable forms. And also one more thing about the oligotroph that they possess a very high surface area to volume ratio for the efficient nutrient absorption and can enter the longer period of dormancy to persist during the unfavorable conditions. Okay? Because you have studied that the bacteria that has the means high surface area to the volume ratio and they have the higher one in the bacterial group that possess the high surface area to volume ratio for absorption of the nutrients efficiently. So some examples we can take in that Coelobacter, okay, Pelagibacter. So these all are the marine examples in the India. The significant research has been carried out also in our Siliguri. We have isolated some oligotrophs from the river Mahananda. So including the means Indian Oceans and the because they degrade the hydrocarbons under the nutrient limited conditions. So Acinetobacter from the Himalayan soils capable of producing the novel antibiotics and microbes from the Sundarban mangroves, the ecosystem contributing the geochemical cycling. So these are the main applications of the oligotrophs. So as they can grow in the scarcity becomes increasing concern in the many ecosystems. So understanding the oligotrophs, their adaptation can open the doors to the sustainable solutions and also the innovative technology. So in today's discussion, this one is the opportunity to share our perspective, to share our knowledge, okay? And we have to discuss that how we can leverage their capabilities for the environmental and the industrial applications of the oligotrophs. So now I welcome all our presents lecturers here. So just you can unmute yourself and if you have any research experience or the research insights, you can share. Yeah. So in my lab means in Siliguri means not it was not my PhD topic, but in our lab we have isolated the oligotrophy bacteria from the river Mahananda. So and we isolated it by growing on low nutrient condition that are to a medium. But our objective was to means searching for the means diverse gene cassettes in the class 1 integro. And we have isolated the facultative oligotrophic bacteria. So we have reported in 2013 the PLOS one journal. I think that would I invited the Arvind Kumar to join because he was the research scholar at that time who carried out this experiment, means this project. So now Brinda, Abhishek, Dr. Prasum sir, Madhuri madam. So our finding was that oligotrophic bacteria, we have proved that they are the good source of the novel genes, and we have also reported there the Mar bacterium from the river Mananda who were the oligotrophs one. So now any participants, if you want to share any your research experience, so we will hear from the Dr. Prakash Salamis at the end. So Madhuri, Abhishek, Prasun sir.
Good evening, everybody.
Good
This is
evening.
Dr. Prasad. Good evening, sir. Very unique topic, sir. Even same for me. It is quite new oligotroph, but quite a bit of information you have already mentioned. I'm just curious about this uh, oligotrophs. These oligotrophs. You mentioned, sir, that it can be isolated from arid soils of Himalayan region as well as from rivers. So how easy or hard it is to isolate bacteria from such environment?
Hello.
Yes, Vikram sir, can you hear me?
Mm-hmm.
Mm. So, how easy or hard it is to isolate and culture these oligotrophs?
From which source, sir?
From suppose Mahananda River you worked on, na? So,
Yes, sir. Yeah. Mananda River we have.
mm,
So we use the sir means R2A media. So this one is the specific media for the oligotroph. Okay. And also we have used the diluted means nutrient agar. Also we have used the diluted nutrient agar for the isolation of this. Okay. So we have used only the specific media and which have grown over that R2A media we have selected it. Because they grow on the same environment means temperature and all. Okay. So only way means in place of the either the diluted nutrient agar means nutrient growth or the R2A media we have used.
okay.
I have
And
called that.
sir, yes, sir.
Hello.
Yes, sir, continue. Yes, sir, continue, sir.
Mm-hmm.
Mm. So, basically, these oligotrophs are good source for finding antibiotic producing bacteria,
Yes.
right? Mhm. Okay, okay.
So for the isolation means we can go for the diluted nutrient agar means poor agar. So 10 to the power minus 1 we have gone means use 10 to the power minus to 10 to the power minus 9. Okay. Means diluted water sample also. Means we have collected the sample from the Mananda, then we diluted that. Yes, Prasan sir. So for EG, we have selected only the facultative means oligotroph, not the obligate oligotroph. Okay. So this facultative of means oligotrophs can grow on both R2A agar plates as well as in the diluted nutrient poor agar.
The number you have done. The number you
Hello. Yes, here Swapnalis, it can grow on the R2A media and the nutrient poor agar.
Sir, heart what?
Pardon? R2A. Just 1 minute, Arvind Kumar is joining. Abhishek, Khushi, Ramesh, Chetwani. So, because the R2A is the nutrient limit means nutrient poor media. Okay, so which one grow over this R2A means it is the facultative oligotrophs here.
Okay, so it's basically a kind of selective media
It's
for
a selective
isolating.
media.
Yes. Okay, yes.
Means for the not it's for the oligotrophs we can say the enrichment media because it select means only the oligotrophs can grow over that facultative oligotroph.
Okay.
Or you can for the screening part you can also use that nutrient agar means poor nutrient agar.
So poor nutrient agar means, sir, it will be having less concentration of nutrient growth. So maybe 10 times we can reduce the content of nutrient growth
Pardon?
and add. Sir, poor nutrient agar means 10 times or 20 times less concentration of nutrient
Yeah, we
growth.
have to just prepare it and dilute it.
Okay, okay. And agar as means common like 1.8%.
Yeah. Hmm.
Yes.
Only have to dilute that component that present in the nutrient
Component
agar.
of nutrient. Yes.
Agar will be same.
Okay. And when you were saying, sir, you need to dilute the sample. So for a river sample, flowing river water usually will have less bacterial load. No.
Yes, yes, right.
Yes.
So that we have targeted, no means Mananda River near to hospital. Our
Okay.
municipality hospital is one, Jila Aspatal there.
Mhm.
So because your objective is
Very.
to hello.
Yes, sir. Yes, sir.
Hmm. Because the Arvind Kumar he did PhD on this topic okay, from our lab. So because the objective was to means gene cassettes in class 1 integron. Okay. So basically means gene cassette means which are the antibiotic resistant genes. Okay. So first he used means that to find out the integron class 1 and then what the genes are present in the class 1 integron. Okay. So he reported that several means mar bacteria okay, from the Mahananda river is present in the oligotrophs.
Okay.
I asked him to join, but he is traveling now.
Because sir, this is so new topic actually. I wanted to
Yeah.
it is a very new thing for me.
New topic, and it's difficult to get isolates.
Okay.
Because in river near to the hospital, and will not get means this in the rainy season. Okay.
Hmm. And sir, there is a question in the chat box also that suppose some bacteria grew on this nutrient poor media or the media you were saying, na? So how can we identify that? Okay, these are oligotrophs and not facultative bacteria or something else. Yes, some other bacteria. Means how to identify that these are oligotrophs.
So as we are using the means oligotrophs, means they have no means any other characteristics other than what means search for that who can grow on means nutrient poor condition. So if you are using the R2S, so this one is the nutrient poor, means a poor nutrient means media. Okay, so if any bacterium grow over this R2S, means they can survive in the low nutrient condition. So we can consider the oligotrophs. Okay. Now for the whether it is the facultative or the oligotroph, so if it can also grow over the means nutrient agar. Okay, so it means the faculty not the obligate. Okay, because the who are the obligate means oligotrophs cannot survive in the high nutrient conditions. Okay, but the facultative to some extent they can survive. So that's why we use which one the means nutrient poor agar as well as the R2A to confirm that if both if they can grow on the both, it means they can survive in the low nutrient conditions. Okay, and also if the nutrient means at the high means concentration of the nutrients, if they can survive means the facultative. Okay, so means our lab have selected the facultative because it's easy to work out with the facultative compared to the obligate.
Okay.
So because
And
there
sir,
is no
this also means that if the nutrient is poor, so they will grow
hello.
slowly. So the colony, yes, sir.
Uh-huh.
So if the nutrient is less than these oligotrophs, they will grow very slowly on the agar plate, or we can expect growth within 24 or 48 hours. Yes, a colony on an agar plate.
No, if this is the facultative, so they can grow means to 24 to 40 hours, there will be colony on the plate.
Okay, okay.
Because their requirement is mainly 0.1 to 50 mg of carbon per liter only.
Oh, almost like water.
Even from the oligotrophs from the drinking water is reported. I have gone through a paper. Oligotrophs from the drinking water. I mean they can survive at that concentration of nutrients also. So we'll see there will be the obligate one. Hello. Dr. Alami sir, now we like to hear from you because here
Yes,
no
sir.
one
Actually,
is
today
expertise in this
there are
field.
few participants, and I am surprised what how to bring the participant every time.
How
Yes,
look
hello.
colonies means what kind of morphology we can observe. So there is no any specific morphology of the oligotrophs. Okay.
Halami sir is online, sir.
Yeah.
Yeah, it is actually a very healthy and useful discussion. But as you have pointed out initially that there is a concern with relation to isolation of those bacteria, whom to call exactly the oligotroph, what is the basis
Yes.
of selecting? According to me, even blue-green algae, cyanobacteria, also can be oligotroph
Mm-hmm.
if I have to classify. Because only the sunlight is enough for them, and then your tap water is enough for them to do the photosynthesis and then grow, right? Or why not the autotroph, which we otherwise call it, which grows in the minimal media? Autotroph also be considered as oligotroph, the one which grows in the minimal media, right?
Mm.
So from the, of course, the metabolism point of view, as you told that they will have the slow metabolism, slow growth, you know. So such characteristics are there, then it is easier to identify
Your voice is cracking, sir.
and what are described. Doing the investigation of these bacteria, if we just see the bacterium kingdom, what is the exact role or their usefulness, if that can come, and whether they can be cultivable, in a laboratory condition, and if they are actually producing a useful metabolites, that can be actually interesting topic, I guess, you know, like bioremediation, or then removal the some other toxins
Mm.
from the environment, detoxifications, you know, like this some useful, or if they are producing some novel antibiotics also, you know, all these things, if it is there, I think can be actually useful. But when we see especially the bacteria which grows in nutrient rich media like bifidobacteria or lactobacilli is there, what we see is they're actually limited biosynthetic capacity, you know, because they don't have a very small genome size. If I have to talk with reference to my expertise and then the topic which is being discussed into this discussion, when we are looking for the limited biosynthetic property, ability, so like lactic acid bacteria has the limited, their genome size, if you see, within the average of 2 to 3 MB. Most of the bifidobacteria are within a 2 MB only, right? And that's why we have to actually give them a nutrient rich media like MRS broth, which has almost 11 components, luxury for their luxury and growth. You need all the 11 different media components to be incorporated, and then kept. So from that perspective, if we compare this oligotrophs and the salt solution, and all these things. So from that perspective, how you can actually oligotrophs actually you can classify or describe, and then what are their industrial application, or in the maintaining the biodiversity, or as an indicator of cleanliness, perhaps, right? As you told that even in the drinking water also you can find
Yes,
this
sir. There is one report, sir.
Yes. So what is the role they play there? And then when we consider drinking water, it is always filtered, right? Ultra purified, filtered one.
Mm-hmm.
So whether during filtration, whether we still have those kind of problem, means whether we can have in the filtered water also or not. Because whatever we see, the coliform or E. coli in most of this Bisleri water or Kinley's water because of the contamination.
Yes,
Right,
sir.
because of not following the good sanitary practices, right? But then if you talk about the oligotrophs which are actually found in the drinking water, so are we considering the water which is actually a potable water which is sold, a packed water, or is it only the tap water, right?
Mm-hmm.
So their industrial application or their application in a biodiversity or in the environment, I think that has to be assessed. And what actually the literature tells about these organisms, I think there must be sufficient literature available when you don't need very much nutrients to grow these bacteria, when they can actually grow with the minimum nutrients, that means it is very good, right?
Mhm.
And then if
Yes.
somebody says, "Do you have such microorganism?" I would just say, because blue green algae, of course, just need the salt solution, and then you will get and then you keep them in the sunlight. No, you will get them nice multiplication because they do photosynthesis and they also produces several carotenoids, you know, and all. So several such bioactive substances, antioxidants, you know, phycocyanin, and all these things. So that is also a very important and useful aspect. The next part actually comes with reference to their genome sequencing.
Mhm.
So how many such oligotrophs have been genome sequenced and how is their origin? In the phylogenetic analysis, from they actually how they are originated, or whether they are the primitive bacteria or what, and whether there is still a scope for those bacteria to get the more differentiated. So that is another aspect which is needed to be understand. Hmm. So this is what from my side. My conference person is calling, so I have to attend his call actually.
Okay, thank you, sir.
Yeah, thank you, thank you.
Vikrant sir,
Yes, sir.
Abhishek, you were waiting. I think some Abhishek is there. I hope you were talking
Abhishek.
about this. Yes, he's there in the participants list.
Yes, sir.
Yes, Abhi. Hello.
Sir, I have a question regarding
Mhm.
oligotrophs. If they survive only in less nutrient condition,
Mhm.
uh, can we use them as sensors to check whether the soil has less nutrients or more?
Yeah, we can. Obviously, I think that we can use it as an indicator.
Yes, that was my question.
Yeah, because in the bioremediation, in the water processing, so these are the applications of the oligotrophs.
Oh,
Okay. So mainly we define oligotrophs in the means in terms of the utilization of the carbon source. Okay. Should not be more than the 10 mg per ml.
Sir, is this oligotrophs are in a symbiotic relationship or what?
No, no, not in symbiotic relation. Because they adapted themselves to grow in the low nutrient conditions. So I don't think that symbiotic relationship is needed here. This was your question here.
Sir, my question is actually can we can find these oligotrophs in symbiotic relationship like lichens and fungi and algae like so?
So it is not in my knowledge because we have isolated, means in our lab, isolated in the nutrient agar, okay, and the R2A media. So there was no such means symbiotic relationship means because this can grow, means oligotrophs can grow in the nutrient, means poor nutrient condition. So I don't think that there is a symbiotic relationship any for their survival.
Okay, sir. सर वन मोर क्वेश्चन।
Yes, sir.
अगर न्यूट्रिएंट कम है तो वो ग्रो हो रहे हैं, वो समझ में आया। अगर न्यूट्रिएंट ज्यादा है तो वो टॉक्सिक नहीं होगा उनके लिए?
If the bacteria is obligate oligotrophs, then it will be toxic for them. Means they cannot survive in nutrient-rich condition. But the facultative can survive. Yeah. Just take the facultative anaerobe. Okay. Facultative oligotrophs can survive in the nutrient-rich condition, but obligate cannot survive. Is it clear now, Khair?
यस सर।
So, is there any more question or any discussion? Person, sir, Khushi Ramesh Chetwani. Just introduce yourself. Khushi Ramesh. Unmute and introduce yourself. Charmi PMM A32333. Abhishek, from which institute you are, Abhishek?
Sir, I am from Savitribai Phule, Pune University.
Oh. You are in postgraduate or
What, sir?
you are faculty member or a student?
I have completed my
Opinion of Abhishek.
MSc.
Our MSc
Yes, sir.
student, MSc student, PhD.
I have completed my MSc and qualified SETNET. I am looking for PhD now.
Oh, wonderful.
Yes, sir.
Yeah, now in Pune you have two culture collection centers, so you should be able to actually give your opinion.
Yes, sir. But
No,
I don't know much about it.
yeah, no,
Yes, sir.
whatever you feel about this topic as a
Yes.
microbiology student. I
Yes,
know
sir.
microbiology professor Kapadnis, Dev Purkar.
Yes, sir.
Yeah,
Yes, sir. Devpurkar, sir. He still teaches in our department.
you see.
Yes.
Yes. Then
Kapadni, sir, also came sometimes as a
one more sir was
guest
there. Who
lecturer.
became vice Kapadnis.
Yes, sir.
And
Kapadni, sir.
then uh, he was from biotechnology, I believe.
Your
Give your
voice.
insight about this topic.
Mm. Yeah, sir. As you said, the nutrient lacking conditions help the microorganism. The first thought that came to my mind was we can use it as an indicator.
Exactly.
Yes, to check whether the soil is rich in nutrients or not.
Yes, yes.
Yes, sir, that was my thought, and I asked the same question. Yes,
Sir, his question was that can we use means oligotrophs as indicator
yes, sir,
for
yes.
the means
And you have responded to that also, and I
hmm.
am satisfied with the answer also. So, is it when you has they been using this as also indicator already? Hello.
Means I don't have an idea now, okay, because but this one is one of the application of the oligotrophs that I have studied, okay, but I cannot give you means exact example that where it has been used.
Okay, okay, sir.
But if you go to the literature, this one is the application, one of the applications of means your oligotrophs.
Oligotrophs.
Yeah.
Right, sir.
But right now I have no any example means where it has been applied.
Okay, sir.
Sir,
Yes.
सर वन मोर डाउट कि जैसे हम डिस्कस किया कि ऑलिगोट्रॉफ्स को न्यूट्रिएंट्स की कमी है और सनलाइट पे भी वो ग्रो कर रहे हैं, तो वो ऑलिगोट्रॉफ्स माने गए, तो उसको स्पेस में ग्रो हो सकते हैं, बिकॉज स्पेस में सपोज मून पे मान लीजिए, तो वहां तो सनलाइट है, तो क्या ऑलिगोट्रॉफ स्पेस में ग्रो हो सकते हैं?
I can think that Halami sir answered this one. Hello, sir. Are you there, sir?
Not exactly to elaborately answer, because what we actually look in the space is more related to the gravity, nutrient uptake.
Hmm.
Then there, no, again the metabolism. Yeah, it can be one model system, but what people are actually using more the pathogenic bacteria. I don't know why. What is the reason? Now Shubh, Shubhanshu Shukla also went, and what are the bacteria he has taken? I think somebody should find out. There was actually an article, you know, from the microbiology perspective, how this space mission from the microbiology perspective is useful, right? So the criteria of selecting such a model system, if it is evaluated, then yes, to some extent it can be useful. That is what I can say, right?
Okay, sir. So basically, it can grow if it fit in that criteria.
Yeah, yes.
Yeah.
Dr. Paknikar's article is there, I think, in the newspaper with relation to the space microbiology, and that was there in this microbiology group also. Dr. Paknikar, who was the director of Agarkar Research Institute, Pune, you know. So maybe somebody can actually review that article and then come back and then have a discussion in this group. It is possible.
Okay, sir.
Yes.
Now the space microbiology also is in part of the extremophiles, sir. So I think that is there any question, Abhishek and here?
No, sir.
No, sir.
So now we can conclude, sir. Dr. Alami, sir.
Yes, sure. Please go ahead.
Vikrant sir, we may ask that one last time. Charmi, I think Dr. Charmi is faculty at Saurashtra. We message in the group. Assistant Professor of
Doctor,
Microbiology and Saurashtra University. May I ask if he is available? Then
yes, sir. Charmi PM. I am Assistant Professor of Microbiology, Saurashtra University. So please unmute yourself and please share your any comment if you have and means your insights on this topic, oligotrophs, or any comment you have or knowledge you want to share. Please unmute yourself. Charmi PMM. One new message. And one more thing I want to add that in oligotrophs, most are now means the uncultured strains. Okay, so we have means scope in this means topic we can say oligotrophs. So thank you everyone for joining today's session. Just I have shared one feedback form, just fill out, okay? Because the last week also now due the issuing of certificates. I will issue certificate within 2 or 3 days. I have shared one feedback form, so just fill out it. Yes,
Yes, sir.
this one is, we can use this one for the bioremediation process and also in the water processing.
Yes, sir. That bioremediation topic is of my interest, and
Yeah.
I have also worked on the bioplastic synthesis.
Because there are report, okay, the bioremediation of the pollutant environments, water treatment processes, okay, and one more field that here has asked that astrobiology, that is space microbiology, okay. So there is
Yes,
a,
sir.
because this one is better than others for this study in space microbiology. So thank you everyone for joining today's session. My special thanks to Dr. Halami sir for sharing his
Thank you, everyone.
insights, Dr. Prasun sir
Sir,
for,
one more question, sir.
yeah.
Sir, कंसिडर्ड अ सिचुएशन जहां दो ऑर्गेनिज्म है, जैसे एक ऑलिगोट्रॉप्स है और एक रैंडमली इक्वली मान लेते हैं। सो अगर न्यूट्रिएंट अवेलेबिलिटी कम है तो ऑलिगोट्रॉप पहले न्यूट्रिएंट ले लेगा या इक्वली पहले ले लेगा न्यूट्रिएंट?
कौन पहले लेगा ना?
हाँ सर, हाँ।
So in my view that E. coli will absorb, means if the nutrient condition is reach one or the good nutrient condition, then E. coli will absorb more. Okay, but when after yes, hello.
नहीं नहीं सर, मतलब ज्यादा नहीं है, उतना ही इनफ है, मतलब कि एक ऑप्टिमम रेंज में है कि ऑलिगोट्रॉप्स पहले ले लेगा फॉर देयर ग्रोथ, बिकॉज उनको कम जरूरत है, तो फिर उनकी कॉलोनी ज्यादा मिलेगी या इक्वली की, मतलब कौन ग्रोथ फास्ट करेगा ऐसा?
When in normal condition,
यस सर, यस।
so E. coli will grow, means you will get the more colony of E. coli. Okay, because they are adapted for the slow metabolism. Okay, oligotrophs have the slow metabolism compared to the E. coli.
ओके सर।
Okay, thank you for joining today's session, Dr. Prasun sir, for your interactive session and for your contribution. Means you have joined almost all the our roundtable discussion. Prakash Salami sir has joined all the our roundtable discussion. So I'm very thankful to both our Dr. Prakash Salami sir and the Dr. Prasun sir to make all the sessions highly interactive and insightful. So thank you everyone for joining today's session.
Thank you, everyone.
So much, sir.
Thank you,
Very
sir.
useful and informative session. Thank
Yeah,
you.
because
बरोबर.
we are only 7 or 8, but we have discussed for more than 40 minutes. So this one is the good for us. This
And
one
it
is
was a quite a new topic for me, I think. It was quite a good topic. Arvind sir has joined, sir. Vikrant sir.
तेच आता ते विसर्जन जाऊन आलो मी गाडीतनं उतरलो तर इकडे आलो.
Are you there, sir? Hello.
Vikrant sir, Arvind sir has joined.
नाही याच्याकडे जाऊन आलो आपण यांच्याकडे नव्हतं गेलो. परवाशीच
Sir is busy.
तो पार्टिशनचं म्हणले ते छान पार्टिशन तुमच्याकडचं खूप आवडलं.
Okay, sir, you conclude as you wish.
Okay. So thank you everyone for joining today's session. And I request everyone to join all our sessions so to make it more interactive and more enriched in knowledge. So thank you everyone.
Thank you, sir.
Thank you, sir.